Hello guys, welcome to IB360. Today I'm back with another video and we have already covered basic facts about endoplasmic reticulum, Golgi apparatus, lysosomes in our previous videos. So go check those out first. And now we have come to the last part of endomembrane system, which is vacuoles. Today we will complete vacuoles part, and with this we will end the endomembrane system and we will move on to the other aspects of cytoplasm. So vacuoles, they are non-cytoplasmic areas present inside the cytoplasm which are separated from the latter by specific membranes. So these membranes, these are known as tonoplasts. In this diagram you can see, and these are selectively permeable membrane. So obviously the vacuoles, they would be fluid filled, and so they would be fluid filled vacuoles or vesicles. So they are separated from the cytoplasm. This is a typical plant cell. Here we have the nucleus and this is the endoplasmic reticulum part. So this is the entire vacuole over here. So this is a diagram of a sap vacuole, and we can see the tonoplast, which is a selectively permeable membrane, separates it from the cytoplasm, rest of the cytoplasm. Vacuoles are believed to be formed by expansion and pinching off from ER. This we will see. So vacuoles formation. Vacuoles are formed when vesicles released by the endoplasmic reticulum and Golgi complex merge together. Newly developing plant cells typically contain a number of small vacuoles. So what happens? Usually a number of small vacuoles are occurring. Sap vacuoles, usually they are occurring in animal cells and young plant cells, and in mature plant cells the small vacuoles, they will fuse to form a single large central vacuole which occupies 90% of the cell's volume. So the small ones, they will fuse and form a larger thing. And usually certain proteins are also involved, known as PBs, protein bodies, which are released from the endoplasmic reticulum, and they incorporate themselves inside these vacuoles. So all these protein bodies are arising or synthesized from endoplasmic reticulum, then they are processed inside Golgi apparatus, and then they merge together, fuse with the individual vesicles, and ultimately whatever is formed is due to fusion of small things, small particles. Ultimately these vacuoles are formed. Usually vacuoles, they occupy a much larger space in plants compared to animals, but they are found in both plants and animals. Vacuoles formation, usually this aspect, in this standard we do not deal with vacuoles formation in that much details, but if you are asked, you should know that endoplasmic reticulum and Golgi complex, both of these organelles, which are very important part of the endomembrane system, obviously they are releasing vesicles, and when the cell matures, these vesicles, they fuse with each other and they occupy a much larger centralized space inside the cytoplasm, almost 90% of the cell's volume in case of plants, and then they would be called vacuoles. Okay, vacuoles types. So there are 4 types of vacuoles: sap, contractile, food, or air. Now sap vacuoles, they have the fluid present inside them, which is often called sap or vacuolar sap, and the composition of the sap, if I have to write it, okay, let me write it here. So composition of sap. Okay, now actually this this particular fluid is extremely hypertonic with respect to the surrounding ECF. The first thing which you must know is the presence of mineral salts, sugars, amino acids, proteins, and also waste products from the cell itself, waste products. Okay, so these are the primary constituents of the sap. Some crystalline deposits may also occur, but not always. Another very important constituent of this sap, they are the water soluble pigments known as anthocyanins or anthocyanthins. Okay, so wait. Water soluble pigments. So water soluble means they can dissolve in water. Water soluble pigments known as and anthocyanthins. Very important. These names are really very important. Okay, so water soluble pigments, they're providing coloration to the cell. Okay, the water soluble things, they are providing coloration to the cell, and the most common water soluble vacuolar pigments would be anthocyanins, which are usually red, blue, or purple, imparting colors, imparting pigments, and anthocyanthins, they are imparting ivory to deep yellow color. Okay, so let me, they are providing with blue, red, purple colors to the cell, because they would obviously impart some colors, and they from ivory to deep yellow. Okay, very important constituent of the sap inside the sap vacuoles. Other than anthocyanins, obviously we have the proteins, esters, amino acids, waste products, and some crystalline deposits may also occur, as I said. Now they may also store food reserve like sucrose, and solutes which are present in cell sap, they maintain a proper osmotic pressure in the cell for its turgidity and water absorption. So they will have solutes, and the solutes will maintain turgidity. In the epithelial cells, in case of plant roots, we know that there is endosmosis of water from soil. The capillary water present in soil gets sucked inside the roots according to their gradient, because the the sap inside the plant cells, they are at a much higher concentration of solute with respect to the soil, and that is why endosmosis can occur, and that is the principal force which is driving the inward flow of water in case of root absorption. Okay, another important thing is latex is stored in vacuoles. Then some plant vacuoles, they also have special transport proteins, and that is why the tonoplast, as I said, tonoplast, they are selectively permeable, and they also, the membrane which separates the cell, separates the cell sap from the rest of the cytoplasm, also bears specific proteins which are helping or maintaining the transportation system across the tonoplast between the cell sap and the cytoplasm. So this is enough about sap vacuoles. I've talked about the composition of sap vacuoles, the water soluble pigments found in them, and whatever their function is, their significance. Second comes the contractile vacuoles. They occur in some protistine and algal cells, mostly found in fresh water. So in this diagram you see, this is an example, or this is a diagram of paramecium, and these are the contractile vacuoles. Surrounding the contractile vacuoles are radiating canals. What these canals do, they are nothing but transmitting water which might be laden with waste products from the cytoplasm towards the contractile vacuole. So these are feeding canals, we say. So the feeding canals, they obtain water with or without waste products from the surrounding cytoplasm, and they will channelize them, or rather they will pour them into the contractile vacuole. As a result, the vacuoles will swell up, and this process is called diastole. When the swollen contractile vacuole, this one, when the swollen contractile vacuole comes in contact with the plasma membrane, it collapses, and this collapsing is again systole. So two important phenomena, diastole and systole. This throws the vacuolar contents to the outside, the systole thing. So diastole is a result of intake of the cytoplasmic contents, and systole is a result of throwing out that content back to the cytoplasm. And these contractile vacuoles, they are helping in osmoregulation as well as excretion. And osmoregulation is required in freshwater habitats where water has tendency to enter the living cells, because obviously the sap is in higher concentration, and that is why water tends to enter the cell, causing it to burst. So this can be prevented by throwing the extra water to the outside with the help of these contractile vacuoles. So very important, their two function, osmoregulation and excretion. Then comes food vacuoles. Food vacuoles, they occur in protozoan protists and several lower animal forms, phagocytes of higher animals even, and they are formed by the fusion of phagosome and lysosome. We have talked about this in our previous class actually. So food vacuoles, they are formed by fusion of lysosome with phagosome. So see, phagosomes are formed when the cell is intaking food particles, the cell eating, through the cell eating process. And lysosomes, they contain the hydrolytic enzymes which are used to digest these food particles. So inside a food vacuole, quite obviously, digestion is performed. So the food vacuole, they contain digestive enzymes with the help of which nutrients are digested, and the digested materials, they pass out into the surrounding cytoplasm. Last is air vacuoles. These are also called gas vacuoles, and they are found only in prokaryotes. Till date, they have been reported only in prokaryotes, the gas vacuoles. Okay, let me write it. So gas vacuoles or air vacuoles reported only in prokaryotes. And another important thing is, we tend to have this idea that gas vacuoles, since they would be light, so definitely they will provide buoyancy to the cell, but they have also other functions to perform. An air vacuole is not just a single entity, neither it is surrounded by a common membrane. Usually we see in case of sap vacuole, we have the tonoplast. Also in case of contractile vacuole, we have some sort of a membrane, a definitive structure. For food vacuoles also, you see here are the food vacuoles. We have a definitive membrane separating them from the rest of the cytoplasm. In case of gas vacuoles, we don't have such membranous organization. It consists of a number of smaller submicroscopic vesicles, and each vesicle would be surrounded by protein membrane, and they would enclose metabolic gases like CO2, O2, and they, in the process, these vesicles, they would be providing us with buoyancy, also with mechanical strength, and very importantly is protection from harmful radiation. So these are the functions of gas vacuoles. So with this we have discussed the various types of vacuoles. We have talked about sap vacuoles, their composition, their function, the water soluble pigments present in them, the fact that they impart coloration to the cell, what colors, we have talked about them. Also about contractile vacuoles, these two phenomena that they undergo, uh, their functions, osmoregulation and excretion. We have talked about food vacuoles, nothing but lysosome and phagosome fused with each other. Their function is digestion. Then we have talked about air vacuoles. These are their functions. Okay, so this is all about vacuoles. In the next class, we will start with the next organelles. Till then, stay safe and like the video, share and subscribe our channel IB360. Thank you.